students, fourth semester students, today we have the tissue culture lecture number 3. It's an important part to have this class in application of plant tissue culture in pharmacognosy. So students should know that when they ask us an essay question, simply if you write the application of various plants development, it will not be sufficient. You should write in detail, in point by point, how it is applied. With examples you should specify. So if you have a basic knowledge about the tissue culture or plant tissue culture, then it is possible for you to write in detail, very simple, and you will get the full mark. I have made this class in an examination point of view, how a student can remember and to write, even if they ask in an essay question, just an application of plant tissue culture. And the students can refer more and increase your knowledge according to the tissue culture. This is just only an outline of application. But the innovative program and the latest technology in development of new drugs and new vaccines, it's needed for you to know about biotechnology application by plant tissue culture. Application of plant tissue culture. The student should know about number 1, the application is production of phytopharmaceuticals. This production of phytopharmaceuticals itself, there is 4 divisions you have to discuss. How, which plants, in what culture. At least a few examples should be specified. We will see in detail. And number 2, micropropagation. Number 3, synthetic seeds. Protoplast culture and somatic hybridization. Number 5, hairy root culture. Number 6, cryopreservation. Number 7, tracing the biosynthetic pathways. Number 8, generation novel compound from plant. Respiration, organ function and metabolic studies. Number 10, plant improvement by studying diseases of plants. 11, mutant cell selection is done by addition of toxic substances. Number 12, production of economic value chemicals. Still there are many innovative programs, development of vaccines, various biotechnology applications are there in plant tissue culture. Number 1, production of phytopharmaceuticals and secondary metabolites, which is the main medicinal active constituents, active principles which we are depending in the nature of the plant sector. The secondary plant metabolites like alkaloids, terpenoids, flavonoids, lipids, oils, tannins, anthraquinones, flavones, naphthoquinones, vitamins, proteins, anti-cancer agents, antiviral agents, and many are isolated from plant tissue culture. Some examples which I have mentioned, that is androquin glycosides from plant species Cassia angustifolia in callus culture. Caffeine, plant species from Coffea arabica in callus culture. Cardenolides from Digitalis purpurea in suspend culture and callus culture. Codeine, Papaver somnifera from suspension culture. Diosgenin, Dioscorea compositae from callus culture. Glycyrrhizin, Glycyrrhiza glabra from suspension culture. Papain from Carica papaya from callus culture. Reserpine, Enravolfia serpentina by suspension culture. Rosmarinic acid, Coleus bloomi by callus and suspension culture. Trigolin, Trigolina phonium graecum by suspension culture. Vincristine, Catharandrus roseus in callus culture. Visnagin in Ami visnaga by suspension culture. Xanthotoxin in Ruta graveolens by suspension culture. So these examples at least can be specified. And in production of phytopharmaceuticals, we can see, that is general examples, we can see again 4 subdivisions of different action in plant tissue culture with production. How? One is biotransformation. Number 2, plant cell immobilization. Number 3, C, genetic transformation. And D, elicitas. These 4 giving various action and helping in the production of various phytopharmaceuticals. How it can be controlled in tissue culture? When you write application, this application subdivisions should be specified. Simply writing biotransformation means transferring from one formula to another formula, or a structure from one structure to another structure, one form to another form, will not be sufficient. As a pharmacist, as a technically qualified person in tissue culture and biotechnology, at least technical terms should be used for explaining these terms. First, a biotransformation, biochemical conversion, that everybody knows. It is a process through which the functional group of organic compounds are modified by living cells. That's why bio. Substrate is converted into chemical different product by chemical conversion by living culture. So this process can be done by using microorganism or plant cell suspension, hair root culture, and immobilized cell. Immobilization is a very important process which you should know. And biotransformation, various terms which comes under various reactions which can give this change of the organic compound or modification of the organic compound in living cell. Biotransformation by plant cell culture yield a wide range of reaction such as glycosylation, glycosyl esterification, hydroxylation, oxidoreduction, hydrolysis, epoxidation, isomerization, methylation, demethylation, and dehydrogenation, etc. So these all reactions we know very detailedly, but it is needed to specify, and all if you don't know, it's needed for the students to know what's these reactions. As we are chemistry background and biochemistry background, it's necessary for the student not simply writing biotransformation. Few examples of biotransformations are as follows. Alkaloids, codenone as a substrate, a product can be obtained, codeine, in Papaver somniferum. This is a biotransformation product by substrate in the plant source of Papaver somniferum. If you specify, the students are getting understanding by the production of a phytopharmaceuticals in the reaction of biotransformation in plant tissue culture. Phenolics, example, rasosinol, glucoside product which is obtained from this substrate in Datura, insomnia, inoxia. And steroids, digitoxin, the product from digitoxin substrate to digoxin in digitalis lanata. Phenolic flavonoids, licuritigenin is a substrate which can give glucoside in Datura, inoxia, and Catharanthus roseus. Phenolic androquinones in ruin as a substrate, and a product obtained is glucoside. Prilla fruticensis. And phenolic coumarins in umbelliferone as a substrate. Into umbelliferone 7 glucoside as a product in Datura, inoxia, and Catharanthus roseus. Terpenoids, citrol as a substrate, a product biotransformation, citrol can be obtained in Lavandula angustifolia. So these are some of the plants which is example in biotransformation experiments which is specified. Even you can find some more examples, and you can find the doubts in these examples also will give you more idea about the biotransformation experiments performed for the production of various phytopharmaceuticals. Second one, in the production of phytopharmaceuticals, plant cell immobilization. Immobilization, you should know, the student should know, is a beneficial aid for production of secondary metabolites, phytopharmaceuticals in plants. Gel entrapment has been the most widely used type of immobilization method. You can see this picture. Immobilization can reduce the hydrodynamic stress, which is putting a stress or a inhibition towards the enzyme for the development and the production of the cell. You can see that cell wall division, expansion in aging, the secretion of alkaline phosphate, SEAP, has been suppressed by means of hydrodynamic stress. So, but in the same time, when you go for immobilization, giving a solidification, a coating film of a gel, which takes place aging, the stress by hydrodynamic stress is reduced. In the arrow have been cancelled. And so that the production of enzyme, the stress is reduced, hydrodynamic stress is reduced by means of immobilization, and the proteases are increased, or the secreted alkaline phosphatase enzyme. In this method, plant cells are fixed in or on a supporting material, or a gel or a matrix such as agar, agarose, calcium alginate, glass, or polyurethane foam. So these are some of the examples which can be used for immobilization. Gel, matrix, which you should know. It involves the entrapment of cell within a gel or passive adsorption on solid support materials, thus creating a situation for cell to imitate membership on a tissue of a whole plant. The three, that is C, genetic transformation, transgenic plant, genetic transformation. The plant obtained through genetic engineering containing a gene, usually from an unrelated organism. Such genes are called transgenes. And the plant containing transgenes are called as transgenic plants. Application of genetic transformation, the gene have been successfully transferred to many crops for resistant to, for what? Various biotic stress. Gene resistant to abiotic stress like herbicide resistance can be given to the plant. Resistance against viral infection can be given to the plant. Genetic transfer to improve quality of the food products. Male sterility and fertility restoration in transgenic plants. Transgenic plants have both basic and applied role in crop improvement. Examples like tomato, tobacco, soya bean, shatavari, papaya, licorice, neem, etc. So this makes the person to have an idea, application of the tissue culture by production of phytopharmaceuticals also. The genetic transformation can give that phytopharmaceuticals which is produced in them can give various resistance and various protection to the plant. Elicitas, this also the fourth one, the D, in the production of phytopharmaceuticals, the coming a division of elicitas. They may ask a short, what is elicitas? So the student should know that. Elicitas are organic or inorganic agents used in tissue culture to trigger rapid and increase the production of secondary metabolites. See, come to the point. Secondary metabolites or phytopharmaceuticals or rapid growth of plant cell culture. So you can see elicitas are organic or inorganic agents. Induction of stress in the plant culture in terms of specific environmental, physiological and biological conditions to enhance the production of secondary metabolites or phytopharmaceuticals is known as elicitation. Elicitas will cause elicitation. The secondary compounds synthesized and accumulated in response to such conditions are called as the secondary metabolites or the phytopharmaceuticals or phytoconstituents which is produced, synthesized in the plant by means of this elicitas or elicitation, which is called as phytoalexins. They are defensing agent in invading pathogens to the plants. So internally they will protect the plant by means of elicitation. They can produce the phytoalexins which can act as a defensing agent in invading pathogens to the plants. The signal triggering in the formation of phytoalexins are called as elicitas. So elicitas definition, you can have a clear cut, detailedly you can give. So you see an example of the picture in a wheat production of phytoalexins. The function of a pathogen have been reversed by means of the phytoconstituents or the phytoalexins produced by the elicitization of the wheat, which all the compounds like phenylamide phytalexin in wheat, like N-cinnamoyl hydroxy 8-oxytryptamine, N-cinnamoyl 8-oxytryptamine. These all are the compounds which are responsible as a phytoalexin in wheat, which can defend the invading pathogen as shown in the picture. So they are produced by elicitas, by elicitation. So you can write very clearly what is an elicitor. And in elicitas also you have two types, very simple, like abiotic, biotic. They have elicitas, abiotic chemical, and biotic they have abiotic chemical, physical. In chemical you can see heavy metals like silver, cadmium, copper and others, ozone zones, ozone, pH control. These all are chemical controls can be done. In physical you can see UV radiation, osmotic stress, salinity, salty, that means salinity means salt content, saltification, salt content, temperature, stress, ultrasound. They are physical abiotic elicitas. When you come to biotic, endogenous, exogenous. Endogenous cell wall fragments like oligosaccharides and protein fragments. Plant hormones, jasmonic acid, salicylic acid, ethylene. When it comes to exogenous, complex yeast extract, fungal spore, bacterial lysates, carbohydrates like cytine, cytosine, dextran, guargum, volatile oils like hydrocarbons, proteins like cellulase. So you see, elicitas, they produce protection for the plants by means of elicitization. They can produce the phytoconstituents which can prevent the plant from various pathogenic attack. And in that they can divide into abiotic and biotic, chemical, physical, endogenous, exogenous. By this, it's needed for you to know about the different types of biotic elicitas. They are derived from microorganisms or produced within the plant cells by plant defensive process against microbial infections. They include mainly beta-linked glucans, cytosine, enzymes, cell wall derived polysaccharides like pectin, pectic acid, cellulose, etc. These elicitas, when added to medium in low concentration, 50 to 250 microgram per liter, enhance the metabolite production. And abiotic elicitas, just now we have seen in the picture, the product accumulation also occurs under stress caused by physical and chemical agents like UV low or high temperature, antibiotics, salt of heavy metals, freezing and thawing cycles by heating different, but example freezing and taking outside, changing the temperatures, conditions, environmental non-essential components of media like agarose, tin, and agaropectin. Certain chemicals like methyl jasmonate, copper sulfate, silver nitrate, etc. and high salt concentration grouped under abiotic elicitas. Elicitas stimulated the accumulation of secondary metabolites or phytoconstituents, phytopharmaceuticals in different plant cultures like opium, Dioscorea, datura, vinca, capsicum, carrot, etc. Micropropagation, cloning, the second application. We have discussed already the micropropagation, cloning propagation. Micropropagation or cloning propagation is a field dealing with the ability to generate plants directly from explants or from a single individual by asexual reproduction constitute a clone. For example, this picture you can see explant in aseptic manner. You can develop callus culture and from that you can see plantlets can be produced from which adult plants can be produced. Micropropagation enables multiplication of sexual sterile and disease-free hybrids. These superior hybrids can be cloned rapidly during this technique. Crop improvements by producing disease-free plants, rapid vegetative multiplication of ornamental plants can be done. Number 3, synthetic seeds, artificial seeds or somatic seeds or synastic or synseeds. Synthetic seeds are defined as artificial encapsulated somatic embryos, shoot but cell aggregates or any other tissues that can be used to showing the seeds and that are having the ability to convert into a plant under in vitro or ex vitro conditions and that retaining the potential also after storage. This is an example for artificially wheat seeds and also artificial seeds for other plants are also available. Various plant species which are reported for artificially seed production are carrot, alpha, somatic hybridization, banana, cardamom, shoot buds and shoot tips, eucalyptus also used in aerial buds, etc. Protoplast culture is the fourth application which we have already seen in the last class itself. Protoplast isolation plays the major role for mechanical isolation and enzymatic isolation. You have to remember about that. This is an application which we have done. Protoplast are plant cells with a plasma membrane but without cell wall. Because of this, the protoplast provides the starting point for many of the technique of the genetic manipulation or manipulators of plants, in particular the induction of somatic clonal variation, somatic hybridization and genetic transfer. They are cultivated in liquid as well as on solid media. Isolation of protoplast are by two methods as we have discussed, that is protoplast can be isolated for almost all plants, that is roots, leaves, fruits, tubers, root nodules, endosperm, pollen cells and cells of callus tissue also. One is mechanical method, second one is enzymatic method. In mechanical method you can see tissue plant tissue will be taken. Cell plasmolysis will take place by mechanically microscopic observation of cell and cutting cell wall with knife. Release of protoplasm will take place without cell wall and collection of protoplasm, mechanical method. And in enzymatic method, leaf sterilization, removal of epidermis, filtration through neon mesh, macroenzyme peptinase in 13% of mannitol. Isolated cells will be added with 2% of cellulose, and 1.5 hour will be half an hour will be kept. Cellulose and pectinase will be added, and 2 to 3 hours for 20 to 22 degrees centigrade, and protoplast can be available without cell wall. Very enzymatic method. Now somatic hybridization after the protoplast, the technique hybridization, hybrid production through the fusion of isolated somatic protoplast under in vitro condition, the subsequent development of their product to a hybrid plant is called somatic hybridization. Various stages of somatic hybridization as follows: isolation of protoplast may be enzymatic or mechanical, fusion of protoplast to the desired species of varieties which you interested, identification and selection of somatic hybridization cell, culture of hybrid cells, regeneration of hybrid plants. Number 5, application, hairy root culture. There are different types of culture, but hairy root culture is a special application. Hairy root culture, also called transformed root culture. It is a type of plant tissue culture that is used to study plant metabolic process or to provide valuable secondary metabolites or recombinant proteins, often with plant genetic engineering. As shown in the picture, it was an interesting study of hairy root culture. Advantages of hairy root culture: genetic and growth kinetic stability or kinetic stability over prolonged period of growth in vitro, easy of culture in vitro using simple media lacking phytohormones, a diversified range of plant species has been transformed using various bacterial strains, plant generation, regeneration, plant improvement and genetic manipulation in plant can be done, reproducible, predictable levels of product synthesis, capabilities of synthesize novel secondary metabolites specific to the plant species from which they have developed in equal to even higher amount compared with the field grown plants, more accumulation of secondary metabolites, that is, example, levels of steroidal alkaloids, psilocybin is significantly higher in hair root culture than when you are doing in a callus or suspension culture. Now, cryopreservation. Immediately you should remember about the nitrogen, liquid nitrogen, negative 196 degrees centigrade, that you should remember. The preservation of cell, tissue and organ in liquid nitrogen is called cryopreservation. And the science pertaining to activity is known as cryobiology. It's called cryobiology. Cryopreservation is a non-lethal. The cells should not be dead. Non-lethal storage. Only you are making it to be statue or to stand or to stay still. Non-lethal storage of biological material at ultra low temperature. At temperature of liquid nitrogen minus 196 degrees centigrade, almost all the metabolic activity of the cell are ceased, made still, and the sample can then be preserved in such a state for extended period, but not for all cells, all plant cells, it's not possible. However, only few biological materials can be frozen at 196 minus 196 degrees centigrade without affecting the cell viability. Without affecting the cell viability, non-lethal storage. Very important, these words. Cryopreservation of few endangered medicinal plants, example Dioscorea, Chirata, Podophyllum, etc., is done by these methods. On the days, there are many, but still a small example, and I just pushing to the next point. Number 7, tracing the biosynthetic pathways of secondary metabolites. Biosynthetic pathway study by means of labeled compound, isotopes. Tissue culture can be used for tracer techniques, tracing the biosynthetic pathways of secondary metabolites using label preservation or precursor in the culture medium. This is a long procedure. There is very lot of examples, but you will be studying about tracer techniques in the next semester. So we are giving just a point which can be applied, but it's a very, very interesting application in tissue culture. Generation novel compounds from the plants, novel compounds. By various methods of plant tissue culture, isolated of novel compounds, liposomes, phytosomes, improvement in yield of existing compound can be achieved. Various compounds can be produced for various release of the drugs. Study, natural release. The latest novel drug delivery systems can also be done in this technique by means of using plant tissue culture and biotechnology. Number 9, respiration, organ function and metabolism in plant tissue culture can be studied. Plant improvement by studying diseases of the plant and their elimination, how the disease can be eliminated with the help of plant tissue culture. Resistance can be developed as we have discussed before. And number 11, mutation and selection of mutant cells is done for significant contribution to a new genetic variability. New genetic variability in the plant can be developed. Number 12, production of economical value chemicals, very, very important chemicals, producing chemicals by plant tissue culture, by even example of insulin itself, you can see, by biotechnology, by plant tissue culture, which are not possible to with other chemical method, can be developed by new researches can be done for making new chemicals to increase the economic value by supplying the need of the medicines and chemicals for the mankind. Application of plant tissue culture, to be remembered, some of the points which we have discussed today is a conclusion, and I want to make it very clear because they used to ask in an essay question. Students should not forget these 12 points, and then in between the first point divided into 4. First one is production of phytopharmaceuticals, which is having 4 different points also. And number 2, micropropagation, which we have discussed. Synthetic seeds, protoplast culture and somatic hybridization, isolation of protoplast, mechanical, enzymatic, you have to remember. Hairy root culture, how it is important. Cryopreservation, don't forget the liquid nitrogen temperature, minus 196 degrees centigrade. 7, tracing the biotechnology, biosynthetic pathway, tracer techniques, labeled compound, isotopes, generation of novel compounds from the plant, respiration, organ function and metabolic studies, plant improvement by studying diseases of plants, mutant cell selection is done by addition of toxic substances, production of economical value chemicals. And in number 1, you should remember about the 4 points: biotransformation, various reactions, and plant cell immobilization, how it can be done by gel, what all the compounds which is used, and see genetic transformation, like you should write about the transgenic plants, elicitas by abiotic elicitas, biotic elicitas, you should write about the elicitas, how it can have the prevention towards the various pathological attack towards the plants can be prevented by the production of phytopharmaceuticals or the secondary metabolites of the plants. So this is what I have given some points which I can give. You can elaborate it by means of various references and do more reference and prepare your notes and listen to the class and make use of it. Thank you for being with me for this application of plant tissue culture.